Hello all dear students, I Mrs.Kiran Y.Bhokare, currently working as an assistant professor in Department of Chemistry at Sinhagad College of Pharmacy, Vadgaon, Pune, delivering a presentation on Denovo synthesis of fatty acids or palmitic acids, which comes under the subject biochemistry. So in today's presentation, we're going to learn about the sites of fatty acid synthesis and steps involved one by one in the fatty acid synthesis in detail. So the majority of fatty acids required for the body is supplied by the diet, and the fatty acids are synthesized whenever there is calorie excess in the diet. Excess amount of carbohydrate and protein from the diet can be converted to fatty acids and stored as a triglycerols. Then the sites of fatty acid synthesis are liver, kidney, adipose tissue and lactating mammary glands. Then it takes place in cytoplasm of the cell. It is referred to as an extramitochondrial or cytoplasmic fatty acid synthesis system. The majority of fatty acids synthesized de novo, that means newly, is palmitic acid, the 16-carbon saturated fatty acid. Source of carbon atoms is acetyl CoA. Source of reducing equivalents NADPH. Source of energy is ATP. Finally, the steps involved in the fatty acid synthesis are 3. The fatty acid synthesis occurs in the following 3 stages. First is the production of acetyl CoA and NADPH. Second, conversion of acetyl CoA to malonyl CoA. And third, reaction of fatty acid synthesis complex, or you can say FAS complex. Then first, production or transport of acetyl CoA and NADPH. As we know, acetyl CoA and NADPH are the prerequisites for fatty acid synthesis. Acetyl CoA is produced in the mitochondria by the oxidation of pyruvate and fatty acid, degradation of carbon skeleton of certain amino acids, and from ketone bodies. Mitochondria, however, are not permeable to the acetyl CoA. So an alternative or a bypass arrangement is made for the transfer of acetyl CoA to cytosol in the form of citrate. So we'll see in this cycle where, as we had seen, acetyl CoA is produced by the oxidation of pyruvate and by the degradation of carbon skeleton of fatty acids and degradation of carbon skeleton of certain amino acids and from ketone bodies. So as we know, the mitochondria is not permeable to acetyl CoA. An alternative or bypass arrangement is made for the transfer of acetyl CoA to cytosol. So here the acetyl CoA condenses with the oxaloacetate in the mitochondria to form a citrate, and citrate is freely transported to the cytosol. Citrate is freely transported to the cytosol where it is cleaved by the citrate layers to liberate acetyl CoA and oxaloacetate. So oxaloacetate in the cytosol is converted to malate, and the main source of NADPH for fatty acid biosynthesis is HMP shunt. So malic enzymes, here the malic enzymes converts malate to pyruvate. NADPH and CO2 are generated in this reaction. Both of them are utilized for fatty acid synthesis. Then second, formation of malonyl CoA. Acetyl CoA is carboxylated to malonyl CoA by acetyl CoA carboxylase. The ATP dependent, and this step is ATP dependent, and biotin is required for CO2 fixation. Acetyl CoA carboxylase is the regulatory enzyme in the pathway. Then here we can see the reaction where the acetyl CoA gets converted into the malonyl CoA with the help of the enzyme acetyl CoA carboxylase in presence of the biotin, and this step is ATP dependent and requires CO2 for the, and biotin is required for the CO2 fixation. Then the third step, where for moving towards the third step, we have to first know about the fatty acid synthesis multi-enzyme complex. So what is this fatty acid synthesis multi-enzyme complex? This is a fatty acid synthesis multi-enzyme complex, where you can see that this complex is the diagrammatic representation of the model of for fatty acid synthase complex, which is also known as a FAS multi-enzyme complex, is shown in this figure. So here you can see this model is tentative and is largely based on the work of Vakil. Fatty acid synthesis is a dimer composed of two identical subunits, monomers, each with a molecular weight of about 240. Each subunit contains the activities of 7 enzymes of FAS and ACP with 4-phosphopentadiene, the SH group. The two subunits lie in the antiparallel head-to-tail orientation. The SH group of phosphopentadiene of one subunit is in close proximity to the SH of cysteine residue of the other subunit. Each monomer of FAS contains all the enzyme activities of fatty acid synthesis, but only the dimer is functionally active. This is because the functional unit consists of half of each subunit interacting with the complementary half of the other. Thus, the FAS structure has both the functional division and subunit division. The two functional subunits of FAS independently operate the synthesis and synthesize two fatty acid simultaneously. So this is nothing but the description about the fatty acid synthase multi-enzyme complex. Okay, then reaction of fatty acid synthase complex. This is a very important part, so we'll see the description in case of a cycle only. So here the fatty acid synthase, this enzyme complex is represented as shown in this figure, where the CYS stands for the cysteine and ACP stands for the acyl carrier protein. Okay, so where, as we know, as we had seen, this fatty acid synthase complex is a multifunctional enzyme complex known as a fatty acid synthase complex. In eukaryotic cells, including man also, this fatty acid synthase exists as a dimer with the two identical units. Here also you can see. Here in the previous slide, as we had seen that each monomer possesses the activities of 7 different enzymes and acyl carrier protein bound to the 4-phosphopentadiene unit. Fatty acid synthase functions as a single unit, catalyzing all the 7 reactions. Dissociation of the synthase complex results in the loss of the enzyme activities. So this is nothing but the structure of fatty acid synthase. Okay, so we'll see one by one the main reactions of fatty acid synthase complex. So here the fatty acid synthase complex. So here in the first step we can see that the 2 carbon fragment of acetyl CoA, 2 carbon fragment containing compound acetyl CoA is transferred to SAP of the FAS synthase complex catalyzed by the enzyme acetyl CoA transcalase. Okay, so the acetyl unit is then transferred from ACP to cysteine. So the acetyl CoA ACP, the acetyl unit is then transferred from ACP to cysteine residue of the enzyme. Thus, the ACP site remains vacant. That means here in this step, the ACP containing the acyl group, acetyl as ACP, that transfer the group of your acetyl unit to cysteine in this step, where the enzyme malonyl CoA ACP transacalase transfers malonate from malonyl CoA to bind the ACP, so that here the enzyme malonyl translocase catalyzes the reaction where malonyl CoA group is attached to the ACP of your FAS synthase. So here then the acetyl unit is attached So again we will see the summary on this slide that this is a structure of fatty acid synthesis where the addition of two carbon fragment containing compound acetyl CoA added to the SCP, that is acyl carrier protein of your FAS complex, where there is a formation of acyl SACP, and then again in the next step there occurs the transfer of acetyl to cysteine, and then again the addition of malonyl CoA going to attach the malonyl group to the ACP of your FAS complex to form the compound acyl manolin enzyme. Okay, so the next slide, the acyl manolin enzyme, the reaction is catalyzed by the beta ketoacetyl synthase where the acetyl unit attached to cysteine is transferred to the malonyl group which is bound to the ACP. The malonyl moiety, that is the malonyl moiety which is on the last slide, that is this is the malonyl moiety loses CO2 which was attached by the acetyl CoA carboxylase. Okay, so here the malonyl moiety which contains CO2 loses this CO2 which was added by acetyl CoA carboxylase. Thus the CO2, here we can see the removal of CO2 from that ACP, so that the CO2 is never incorporated into fatty acid carbon chain. The decarboxylation is accompanied by the loss of free energy which allows the reaction to proceed forward, and this reaction is catalyzed by the beta ketoacetyl ACP synthase. Okay, then after removal of the CO2 we'll get the beta ketoacyl ACP. In the next step, the beta ketoacyl ACP reductase reduces ketoacyl group to hydroxyacetyl group. So here the ketoacetyl group is reduced to the beta hydroxyl ACP. Okay, so here occurs the reducing equivalents are supplied by the NADPH. Okay, then beta hydroxy acetyl ACP by the removal of a water molecule or undergoes the reaction of dehydration, a water molecule is eliminated and a double bond is introduced between a alpha and beta carbon. Here between the alpha and beta carbon the double bond is going to get incorporated. Okay, so some summary of reactions in this slide is, as we know, the removal of C double bond O from the acyl manolin enzyme, there occurs a formation of beta ketoacyl ACP in presence of the enzyme beta ketoacyl synthase. Okay, then again after that the beta ketoacyl ACP is converted into beta hydroxyacyl ACP with the help of the enzyme beta ketoacetyl reductase. Okay, where then again into the last step in this presentation, in this slide, there occurs a reaction of dehydration, that is removal of water molecule in presence of the beta hydroxyacyl dehydratase to form the transenol ACP. And then again transenol ACP, a second NADPH dependent reduction catalyzed by enol ACP reductase occurs to produce acyl ACP. The 4 carbon unit attached to ACP is butyryl group, that is here the 4 carbonyl group attached here is the butyryl group, and then again there occurs the formation of acyl S enzyme by the reaction of transfer of carbon chain to cysteine SH. Okay, and then 6 more cycles of reactions from step 1 to step 6 occurs where there is formation of acyl ACP takes place, and this reaction is catalyzed by the enzyme thioesterase